Hello everyone, my name is Jana Abasbe and today I'll be presenting my bachelor's thesis on higher order adaptive modeling for the role of epigenetics in Huntington's disease. I've developed a computational framework to understand how the environment can influence the progression of the disease through epigenetic mechanisms. And so let's get started. I'll cover 5 main sections today. First, a brief introduction to the project and some background on Huntington's disease. Then I'll introduce the important role of epigenetics in its development. After this, I'll explain my higher order network design approach and show you the specific adaptive model I created. After that, I'll demonstrate the model through simulation experiments to show both disease progression and therapeutic intervention. And finally, I'll conclude with the main insights and discuss some future research directions. Now for an introduction, in one long sentence, this thesis introduces a fifth-order adaptive dynamical system model to analyze the epigenetic mechanisms linking environmental stressors to the progression of Huntington's disease, or HD as I will refer to it sometimes, particularly focusing on the downregulation of brain-derived neurotrophic factor, or BDNF. BDNF is a protein that's crucial for neural survival in Huntington's disease. So what is HD? It is a devastating neurodegenerative disorder that affects hundreds of thousands of people worldwide. It is characterized by neuron degeneration in the brain, but particularly and most severely in the striatum. And it presents with 3 main symptom categories. So we see primarily motor symptoms like chorea, which is sudden, uncontrolled, involuntary movements, as well as cognitive and psychiatric symptoms, with up to 70% of patients experiencing depression and anxiety. Now, we know the genetic cause for this is a CAG repeat expansion in the Huntington gene, leading to mutant Huntington. But this mutation, the length of this CAG repeat expansion, has been shown to explain only about 56% of variability in the age of onset. This means 44% of the variance that remains in when people develop symptoms needs to be explained by other factors, which has led to a growing body of research focusing on the roles of the environment and epigenetic factors in modulating the onset and progression of Huntington's disease. So what happens to the brain in this disorder? Well, we see how, or I mentioned how Huntington's disease causes progressive neuronal degeneration. And we see it in this diagram here. So in comparison with a healthy brain, we have selective degeneration of medium spiny neurons in the striatum, as seen here. And we also see loss of cortical neurons in motor and cognitive areas. This means also disrupted corticostriatal circuits, and all these changes in the brain lead to the motor and psychiatric symptoms I mentioned just before. Now let's transition to the core of this research, epigenetics in Huntington's disease. But first of all, what is epigenetics? Well, it refers to changes in gene expression that don't alter the DNA sequence itself. So we can think of it as sort of a switch for a gene. It can turn it up or down without changing the actual genetic code. And this is particularly interesting because it's sensitive to environmental influences and it can potentially be reversible. So it offers hope for treatment. Now we're particularly focusing on the epigenetics of neurodegeneration, and this figure from Babenko and colleagues nicely illustrates this relationship between the environment and degeneration of the nervous system. So we can see different environmental factors here and how epigenetics acts as sort of an interface that translates these environmental factors into gene expression. And this epigenetic response in the brain can either delay or accelerate these neurodegenerative processes in diseases like Alzheimer's or Parkinson's, and of course, Huntington's disease. Now for the main focus of my research, BDNF, or brain-derived neurotrophic factor. On the left here, we see some of BDNF's normal functions. So we know that it promotes neuronal health, survival, and plasticity. It is good to note that it's produced mainly in cortical neurons and it's transported to the striatum. And we saw before how these corticostriatal circuits are affected in HD. Importantly, BDNF supports medium spiny neurons, which express higher levels of TRKB. So TRKB is the receptor for BDNF, and the MSNs in the striatum, which express higher levels of this, are the neurons most affected. BDNF is shown to be downregulated in Huntington's disease. We see up to an 82% reduction in the striatum, and it is shown to be reduced also in the prefrontal cortex. This reduction leads to impaired synaptic plasticity and function, as well as degeneration and atrophy of neurons in the brain, especially in the striatum. Now, how does this BDNF downregulation happen? We're going to use this figure from Speidel and colleagues to help us demonstrate this. On the left side here, we see normal neurons, and on the right, Huntington's disease neurons. And here, two key mechanisms are shown. My research focuses primarily on the one below here, and this is DNA methylation or hypermethylation. We see here how environmental stressors, for example, can activate DNA methyltransferases or DNMTs, which methylate elements of this BDNF exon 4. This silences the gene, so we can see how it goes from on to off. Second, here in section A, we see rest translocation. So on the left-hand side here, we see how in normal neurons, wild-type Huntington protein binds to rest in the cytoplasm. And rest is a protein that acts as a transcriptional repressor. And so this binding prevents rest from translocating to the nucleus. And but then we see how mutant Huntington does not bind to rest in the same way. And then rest translocates to the nucleus, and this allows it to form repressor complexes on BDNF exon 2, also silencing the gene. So these two mechanisms converge and lead to BDNF silencing, and we see this BDNF downregulation in action. Now I demonstrated these complex biological processes, and let's move on to the computational approach that I use. This model uses Professor Truer's approach, and this provides the ability to model these complex dynamics across many levels. So structurally, my model consists of 25 states across 6 levels, and these levels represent sort of a biological hierarchy. And let's start at the base level, which is the foundation of the model. In this level, we see behavior, emotional and motor regulation. So this cycle here is the motor regulation cycle controlled by the striatum, and below here we see the emotional regulation cycle controlled by the prefrontal cortex, and I adapt this cycle from Professor Truer's book. Now for a brief overview of the states, we have these context states. Context daily provides normal environmental context, and context stressors is activated during stress periods. Then we have these sensory states and sensory representation states, which model the sensing of a stimulus and the brain's representation, so the brain's interpretation of the stimulus, respectively. And then we have these preparation states for up here an action and down here an emotion. And then, so the striatum can control the preparation and execution of an action, and the prefrontal cortex is able to modulate this cycle of preparing and feeling an emotion. We have these blue arrows that point all the way from the lowest layer to the highest layer. And this represents how the feeling of stress can, so this allows us to model how the feeling of stress can affect DNA hypermethylation, leading to BDNF downregulation. So let's talk about the adaptivity in the model. Every layer above this pink layer is an adaptive one. And adaptivity is introduced through the use of W states. And these W states are self-model states, which represent connection weights. So, for example, this W, PSB, PFC represents the connection between PSB and PFC. And this allows us to model changes in this connection. So let's go through this layer quickly. This is the brain structure layer. It represents changes in synaptic plasticity and neuronal health, particularly in the medium spiny neurons and also in the cortex. Moving up, we have the enzyme layer. This one represents changes in enzymes and proteins, their levels, and how they and these this level modulates the one below, so it affects the health of these circuits on the layer below. Moving up, we have this third order adaptation, which represents mRNA levels, and these mRNA levels influence the enzymes and the proteins in the layer below. Fourth order adaptation shows the expression of the BDNF gene. And finally, above, right at the top here, we have the epigenetic layer, and this shows the DNA hypermethylation, which affects BDNF gene expression. So these effects that are downstream of this DNA hypermethylation are represented through these pink arrows that go all the way down. So this allows us to model how DNA hypermethylation causes BDNF gene loss of BDNF gene expression, lower mRNA levels, decreased protein synthesis, and decrease in neuronal health, and finally the effect of this on symptoms. Right, so this is the conceptual model, the main one, and then we have the therapy model. And the only difference between this one and the one before is the introduction of a context state at the epigenetic level, and this represents DNMT inhibitors. So we saw what DNMTs did before at the BDNF promoter 4, and this context therapy state has a strong positive connection to counteract DNA hypermethylation. So when this state is activated, it facilitates this recovery process that goes all the way down. So we have reduced DNA hypermethylation, which allows the BDNF gene expression to recover. And this leads to increased mRNA, more protein synthesis, improved neuronal health, and finally some restored motor and emotional control. Now that we've explained the model conceptually, we can talk about the computational foundation that makes the modeling possible. So every state Y evolves according to this difference equation, where it changes based on the weighted impacts from input states x1 through xk. And this is modulated by this is aggregated through the use of combination functions, cy here, and the speed factor eta controls how quickly this state y responds to these influences. Now, when connection weights become adaptive, equation 1 transforms to equation 2, and instead of fixed weights, we have these dynamic W states that change over time. So which combination functions did I use? I selected two of these combination functions, the advanced logistic sum and the step once function. The advanced logistic sum provides a smooth threshold effect, so it allows us to control when and how strongly certain influences take place. So the steepness parameter sigma controls how sharply the transition happens from off to on for this biological response. And the threshold parameter tau determines the sensitivity level needed to trigger the response. Now the step once function is a little bit simpler. It provides temporal control. So it takes two parameters, alpha and beta, and these are the start time and the end time. And so this state is active within this time frame and not active otherwise. So how is this implemented? We use the software environment developed by Professor Trur. And this environment can automatically generate and solve the system of n, in my case 25 or 26, coupled difference equations automatically. The entire model specifications, so we have the network characteristics, so the connectivity characteristics, the aggregation characteristics, and the timing characteristics. These are all defined through role matrices. Which states are connected to which in the base connectivity matrix. And then we have these connection weights, the weights of the combination functions, and of course the parameters, and the speed factors and initial values for every state. Once these are specified, the environment integrates the system dynamics, and we can simulate these processes for analysis. Now let me show you how the model performs through two key simulation experiments. So in the first scenario, we have the development of Huntington's disease. So initially from time 0 to 100, we see a normal daily life with this context daily state. But we also see a steady decrease in W states due to the baseline effect of mutant Huntington on epigenetics. So this is the genetic vulnerability. At time 100, I introduce environmental stressors using the step 1's function. And this causes a sharp drop in the W states. So we see what I described earlier with lower BDNF expression or DNA hypermethylation leading to lower BDNF expression, lower mRNA levels, protein synthesis, and neuronal health. And finally, this translates to the symptoms. So we see here in red and in green a spike in the activation levels of the prefrontal cortex and the striatum due to increased sensory input, but then they both drop down during this time frame and they stay low after these stressors are removed. What this means also is that this execution state in blue increases from very low levels to a pathologically elevated level at around 0.7. And this represents chorea, where movements are no longer controlled by the striatum correctly. Emotionally, we also see these preparation and feeling states for emotion are pathologically elevated, and all of these changes persist after the removal of stressors. In the second scenario, we see for the first 400 time units the same simulation as before, but then at time step 400, I introduce the hypothetical therapy using DNMT inhibitors, and these DNMT inhibitors restore BDNF expression and have a positive effect on symptoms. So we see how the DNA hypermethylation no longer causes BDNF downregulation, so BDNF levels increase back to pre-stressors levels, and also the downstream effects of this. So we have the increased mRNA, increased protein synthesis, better neuronal health, and this translates to the improvement of symptoms. So we can see how the striatum and the prefrontal cortex restore some normal function. And the execution state, for example, drops back down to pre-stressors levels, and the feeling state and emotion state also dropped back down to pre-stressors levels. And all of these changes also persist after the removal of the therapeutic intervention. Now let's conclude by highlighting some key insights and future directions of this work. So first, regarding environmental factors, this model can effectively demonstrate how stressors can accelerate Huntington's disease through epigenetic BDNF downregulation. This improves our epigenetic understanding, so a better understanding of the epigenetic contribution to this neurodegenerative processes. And this model also introduces therapeutic potential, or models therapeutic potential. So this therapy simulation suggests that epigenetic therapies can improve established symptoms of Huntington's disease, and this offers hope for treatments. And finally, this model successfully integrated genetic, environmental, and epigenetic factors in one computational framework. Finally, there are a number of research directions I think would be very interesting to pursue building on this work. So to improve the biological accuracy, we can include other processes specifically involved in the selective degeneration of medium spiny neurons. So oxidative stress, protein aggregation, mitochondrial dysfunction. Of course, my model is a simplified model that just treats the downregulation of brain-derived neurotrophic factor, but many other factors are at play here, and it would increase the accuracy of the model. There's also emerging research suggesting that parental stress can influence the severity of Huntington's disease in children through epigenetic inheritance, and I think I would love to look at these transgenerational effects in the future. And finally, for therapeutic modeling, I think it would be exciting to explore therapy on a different biological level. So I explore an epigenetic therapy, but there is research suggesting that lowered stress levels and for example, environmental enrichment in animal models, also improvement of diet can contribute to better disease outcomes, and I think this would be very interesting to model as well. And that's all for me today. Thank you so much for your attention, and I'm happy to answer any of your questions.